Hello, today we'll talk about the fourth topic, which is animal nutrition, and we'll see the different regions of the alimentary canal and what are the enzymes and what are the substrate that used to, what are the enzymes and its substrate to break down the foods into simpler components until do the absorption. For animal nutrition, we need to know the alimentary canal. So the alimentary canal is composed of a different region, as you can see from the figure, and we will discuss these regions and their functions in details. You can see the major regions on the top right figure. You can see where the storage occur in the stomach, where the digestion and absorption occur in the intestine, while the egestion from the anus and the ingestion from the mouth. You can see as well that generally the alimentary canal is lined by multiple many layers, including a lining epithelium with digestive glands and the circular and longitudinal muscle fibers. So basically nutrition is divided into several processes: ingestion, digestion, mechanical and chemical, absorption, assimilation, and digestion. Ingestion, of course, is taking the food substance as food and drink into body through the mouth, while the mechanical digestion is the breaking of food into smaller pieces without chemical change to food molecule, while the chemical digestion is the using of the enzymes secreted by the digestive tract to transform the large insoluble molecules into soluble ones. The absorption is the movement of these small food molecules and ions through the wall of the intestine into blood, while the assimilation is the movement of these digested food molecules into cells of body, where they are used and become part of the cells. And finally, the egestion is passing out the food that have not been digested or absorbed as feces through anus. And from the figure, you can see where is the regions of these processes occur. So if we have this table to see the location and the function of the main parts of the alimentary canal, we can see in the mouse three processes occur: ingestion, mechanical digestion, and chemical digestion. Ingestion, of course, of food using mouth and teeth, lips, and tongue. Mechanical digestion by the teeth. Chemical digestion of starch only occur in mouth by the enzyme amylase. Tongues is used to mix the saliva to form something called bolus, which can be easily swallowed. And then the mouth leads to the buccal cavity, to the throat or pharynx. The salivary gland, which each digestive gland, it forms a digestive juice formed of water, mucus, and amylase. This water dissolves substance in food, allowing us to taste it. Mucus help to stick this chewed food to form the bolus, and for easy lubrication of the mouth and easy swallowing. While the amylase, which called as well ptyalin, is used for chemical digestion of cooked starch and break it down into maltose. So amylase works on cooked starch, not uncooked starch, to convert it to maltose. Esophagus, or the gullet. Actually, esophagus, it only functions for transferring the food from the mouth to the stomach by a process called peristalsis. It is behind the trachea, which is the windpipe. There is an epiglottis, a flap of cartilage cover the entrance and trachea, which prevent the food from entering the windpipe. And it guides the food into esophagus as well by passing over the windpipe. The entrance to the stomach from the esophagus is guarded by a special kind of ring of muscles called sphincter. This muscle relax to let the food pass into the stomach. Once the food reaches the stomach secretes gastric juice, which contain pepsin for chemical digestion of proteins. It produces hydrochloric acid as well to kill bacteria, and the peristalsis mixes food up into a liquid. In the small intestine generally is divided into two regions, duodenum and ileum. Duodenum is the first part, while the ileum is the second part. The duodenum receives the pancreatic juice for chemical digestion of proteins, fats, and the starch, and neutralize the acid coming from the stomach as well. It receives the bile to emulsify fat, and this type of digestion is called physical digestion. Actually, it's converting the fats into smaller chunks of pieces. While in the ileum, the enzymes in the epithelial lining carry out chemical digestion of maltose and peptides. The ileum is very long and has villi to increase the surface area for absorption. The pancreas, it's a mixed gland, secrete pancreatic juice into the duodenum via the pancreatic duct for chemical digestion as well of proteins, fats, and starch. The liver makes bile, contains salts to emulsify fats, and it's another mode of digestion, physical digestion. In liver occurs assimilation of digestive food such as glucose and deamination of excess amino acids. The gallbladder, which is in the liver, stores bile to be secreted into duodenum via the bile duct. While the cecum and the appendix function, the undigested food travel on through the cecum, past the appendix and onto the colon. In humans, the cecums and the appendix up till now have no function. The inside of the alimentary canal is lined with epithelium. New cells in epithelium are being produced all the time to replace the damaged cells by movement of the food, because the movement of the food scratches the lining of the epithelium, so they cast off, so they need to be renewed. Cells in epithelium can produce mucus, and this mucus is important in lubricating the lining of the canal, and it protects it from wearing and tearing. Mucus also protects the lining from the attack of the digestive enzyme, which are released into the alimentary canal. Some of the digestive enzymes are produced by cells in lining of the alimentary canal, as in the stomach lining. Other enzymes are produced by digestive glands that are outside the alimentary canal but pour their enzymes via ducts into the alimentary canal, like of course the salivary gland and the pancreas. Mechanical digestion. As you can see, if we're talking about mechanical digestion, we'll start talking by the teeth and the structure of the teeth. You can see the jaw, how it looks like. There is incisors, canine, premolars, and molars. And at the back of each lower and upper jaw, there are the wisdom tooth, which is actually a molar. And there is an upper jaw and lower jaw, and each teeth has a crown. So this table summarizes the types of the teeth and the position in the mouth and the description, its shape, what looks like, as well as a function. Of course, is the incisors function beating off pieces of food, while canine similar function to incisors, while the premolars tearing and grinding food, while the molars are chewing and grinding food. The big difference between the premolars and molar, that premolars have 2 points cusps and have 1 or 2 roots, while the molar have 4 or 5 cusps and have 2 or 3 roots. The canine have a pointed end more than the incisors. If we look closely into the structure of the tooth, you can see that generally it consists of an upper part called the crown, the part which is visible above the gum line, while the lower part is the root, which is embedded in the jaw bone. In the upper part, the tooth is divided into an enamel, and it's a very hard layer covering the crown, and followed by underneath the enamel there are the dentine, which is softer. These 2 layers enclose a cavity which is called pulp cavity, which is inside the dentine and have nerves and the blood vessels. The tooth is embedded in gum, which is the tissue overlays the jaw. The whole tooth is fixed into the lower jaw, into the jaw's bone by a material called cement, and this enables the tooth to grip to the bony socket in the jaw. Terminally, you can see terminally that the tooth is supplied by blood vessel and nerves to enter the tooth through a small hole at the base of the root. You can see this figure, and you can see a side figure as well for the tooth. So the teeth undergo mechanical digestion or mastication, which, as we told you before, it's a breakdown of food into smaller pieces without any chemical change. Humans are omnivorous, so they have all the types of the tooth of the carnivores, but human teeth are not used for catching, holding, or killing or tearing up prey. Mammals also differ from other animals by having 2 sets of teeth, a milk set, which is deciduous, and the permanent teeth. What's dental decay? Dental decay is caused due to bacteria in mouse. The bacteria in mouse with saliva forms a sticky film over the teeth next to the gum and in between the teeth, and as you can see from the figure, it's called plaque. Plaque is soft and easy to remove at first, but if it hardens, it forms another structure which is called tartar that cannot be removed by brushing, so you need to go to the dentist to clean this structure. The gum disease. Okay, if you can see, if plaque is not removed, bacteria in it may infect gum. The gum start to swell, inflamed, and they may bleed when you brush your teeth. But if the bacteria spread, they may reach the root of the teeth. The tooth become loose and needs removing. As you can see from figure 1, start plaque is forming, then the plaque is not removed, the inflammation reach inside to the gum, and now the tooth is loosened and may fall out to be removed. Tooth decay. If sugar is left on the tooth, bacteria in plaque will feed on it. The decay begins when small holes like cavities appear in enamel, caused by bacteria. The bacteria feed and respire on sugar deposited on teeth and produce acid. These acids dissolve the calcium tooth in enamel, exposing dentine, and dentine is very sensitive because it is connected to nerves, which is inside the pulp. Dentine is softer than enamel and dissolve more quickly, so cavities are formed. You can see from the figure it starts by breaking the enamel. Once reached the dentine, the rate of dissolving is much faster. Then the cavity become distance between the cavities reduce the distance between outside of the teeth and nerve endings. So nerve will sense any change in the mouth. The acid produced irritate nerve ending and causes tooth ache. If the cavity is not cleaned and filled by a dentist, the bacteria will get into the pulp cavity and cause a painful abscess at root, as you can see from figure 4. Often the only way to treat this is to have the tooth pulled out. Now we will talk about chemical digestion. Starts with the mouse taking food that is ingested. The mouse, as I've been told before, only chemical digestion of cooked starch by the enzyme amylase, which is a tiny. The mouse leads to the buccal cavity to the throat or pharynx. Followed by the esophagus, the food is forced down by the gullet by peristalsis to the stomach. The stomach has a strong muscle walls, as we said before. It's called the pyloric sphincter muscle, which is a circular band of muscle at lower end of the stomach that stops solid pieces of food from passing through the duodenum. The main function of the stomach is actually 3 functions: store food, turn food into liquid, and release food into small quantities at a time to the rest of the alimentary canal. This peristaltic action of the muscles in the wall of intestine is a kind of a physical digestion. These muscles contract and relax, squeezing food and mixing it with gastric juice, turning the mixture into a creamy liquid called chyme. So it starts from the mouth called the bolus, and then it's transferred to another structure which is called chyme. So now the chyme will move to the small intestine. Continuing talking about the stomach contains different types of cells. And these cells situated in a pit, like the figure. This is actually a gastric pit in the figure. This gastric pit in the stomach wall which actually secrete the gastric juice. These pit contain important type of cells called goblet cell, as you can see from the figure. These goblet cells secrete the mucus. Other cells which produce the main protease enzyme, which is the pepsin, other make hydrochloric acid. The pepsin, of course, digests proteins into polypeptide, and this pepsin must work in acidic condition. In renin, what's renin? Renin is only produced in the stomach of young mammals to clot milk they got from mother. So the milk protein are then broken down by pepsin. After 1 or 2 hours, the sphincter at the bottom of the stomach opens and let the chyme pass into the duodenum. And you can see from the figure the cells which will produce mucus, then followed by cells which makes pepsin, followed by cells which make hydrochloric acid. And these constitute the cells of the gastric pit. In this figure, you can see the relationship between stomach, liver, and pancreas. You can see where is gallbladder is located. where is the sphincter, the pyloric sphincter is located, and then the stomach is filled, is followed by the duodenum. The hyaluronic acid secreted by cells in the wall of the stomach creates a very acidic medium, reaches a pH 2. This pH is important because of two factors. It denatures enzymes in harmful microorganisms in food, bacteria, so bacteria will die in the stomach due to acidity, and as well it provides optimum pH for the pepsin to work. Let's talk about the small intestine, which are around 5 meters long. The pancreatic juice and bile from liver are produced into duodenum. The pancreas, which is a digestive gland lying below the stomach, it makes a number of enzymes which act on all classes of food. The protease, the pancreatic amylase, which is different from the amylase which is in the mouse. The lipase, the protease here is called trypsin, which breaks the protein into polypeptides and followed by amino acids. While the pancreatic amylase, of course, break down starch into maltose. While the lipase break down fats, lipids into fatty acid and glycerol. This pancreatic juice contains as well sodium hydrogen carbonate, which is particularly important in neutralizing the acid coming from the stomach. And this is necessarily because the enzyme working there in the small intestine must work in alkaline medium and do not work well in acidic condition. All the digested material now is changed into soluble compound, which can pass through the lining of intestine and into the bloodstream. If we were talking about the small intestine, we need to know the structure of the villi. The inner wall of all parts of small intestine, the abduodenum and the ileum, is covered with millions of villi. Each villi is about 0.5 to 1 millimeter long. Cells covering villi make enzymes. The enzymes do not come out into the lumen of the small intestine, but stay close to the cell which make them. This enzyme will complete the digestion of food. The carbohydrate enzyme maltase breaks maltose into glucose. The proteases finish breaking down any peptides into amino acids. Lipase complete the breakdown of fats into fatty acid and glycerol. So the outer membrane of each villi cells have another structure for increasing the surface area, which is called microvilli, which increases the surface area by 20 times the exposed surface to the cell. So each microvilli have on its border, each villi have on its border another structure called the microvilli to increase the surface area 20 times. What is the significance of chemical digestion? Solid food is dissolved to make a solution. This breaking down of large molecule into small molecule to enable it to be easily absorbed. The small molecule can be absorbed through the epithelium of the alimentary canal, through the walls of the blood vessel and into the blood. So amylase in mouse secreted by the salivary gland, while in duodenum amylase come the pancreas. The proteases in stomach it's a pepsin, and in duodenum is trypsin, which is secreted as well by the pancreas. While the lipase in duodenum, lipase secreted by pancreas. Starch is digested in two places in alimentary canal. By salivary gland by salivary amylase in the mouse, and by pancreatic amylase in the duodenum. This chemical breakdown of starch takes place in two stages. Amylase work best in neutral or slightly alkaline pH and converts large insoluble starch into smaller one, soluble 12-carbon maltose chain. The maltose is a disaccharide and too big to be absorbed through the wall of intestine. That's why maltose is broken down to glucose by maltase, which is present in the membrane of the epithelial cells of villi of the small intestine. In this figure, we'll summarize the processes that's already happening. So if you think, the first figure, if you think that this is a large molecule, example starch, it is broken down by enzyme A, which is the amylase. It breaks down into peptides, which is the maltose. This maltose is attacked by a different enzyme and broken down into a single amino acids. The digestion of proteins. There are two major proteases break down proteins, as we said before, pepsin and trypsin. The pepsin is secreted by the stomach, while the trypsin is secreted by the pancreas. The pepsin convert the proteins break down into small peptides, shorter chains of amino acid, while the trypsin have a similar effect and have a similar role to pepsin, but works in the but secreted in the pancreas in the small intestine. The small intestine itself doesn't produce enzyme. We have crepes in the villi, so the villi is not a digestive gland, but can produce mucus. We have the crepes in the villi. These crepes, the main function of rest is actually to renewing the cells that rupture due to the movement of food. So they produce new epithelial cells to replace that lost from the tips of the line due to the movement of the food. The epithelial cells of the villi contain enzymes in their membrane that complete the breakdown of sugars and peptides before they pass to bloodstream. So we have peptidase that breaks down polypeptides and the peptides into amino acids. Bile and its function. The bile actually is a yellowish-green fluid made in the liver, bile pigments. Hemoglobin from RBCs are broken down and pigments are excreted in feces. The bile is stored in gallbladder and delivered to the duodenum by the bile duct. Actually, the bile doesn't contain any enzymes. The bile is alkaline due to the presence of sodium hydrogen carbonate, which neutralizes the acidic mixture comes from the stomach and the gastric juice as it enters the duodenum. Enzymes secreted in the duodenum needs alkaline medium, so that's why there is a sodium hydrogen carbonate for optimum rate of reaction. The bile contains as well bile salts which act on fat like a detergent for emulsification. So this emulsification is a kind of mechanical digestion. They break them into small droplets with a large surface area to be more efficiently digested by lipase to fatty acids and glycerol. In this figure, we'll summarize how mechanical and chemical digestion work together to produce small molecules the body can use. So we either eat fats or carbohydrates or proteins. So the teeth break down large pieces food into smaller one. As you can see, all these raw material have been broken into smaller pieces. The bile salts for fats break down into large drops of fats into smaller ones, while the water in the digestive juice dissolves some of the proteins into small pieces of food and some food in solution. As well, the water will dissolve these carbohydrates into smaller pieces. To continue, the fat now is converted into smaller molecules and the proteins are converted to peptides, while the starch molecules start by the action of all these different enzymes, either lipases, proteases, or amylases, will be broken down into smaller molecules. With the help of the peptidase, the amino acids are formed. With the help of maltase, the glucose molecules, the simpler molecules are formed. So this figure explains how the large complex compounds are transformed into smaller compounds that is easily absorbed, or it explains how the large insoluble compounds, which is protein, carbohydrates, and fats, are transforming into soluble compounds that can be easily absorbed, which are the glycerol fatty acids, amino acids, and the glucose. Okay, this table is a very good one for summarizing all what we have been said in the lecture. It's a summary of digestion in the alimentary canal. You will find all the enzymes we have talked about. You will see which part of canal involved, which juice is secreted, where it's made, the enzyme in juice, the substrate is working on, the product, the final end product, as well as the other molecules in the juice, as well as the function of the other substance. So it's like a summarize for the whole lecture. All the digestive juice must contain water and mucus. The water is used for the digestion of large molecules into small molecules, and it is also act as a solvent for the nutrients and enzymes. Mucus as well act as a lubricant. It also forms a covering over the inner surface of the alimentary canal and preventing enzyme from digesting the cells. Absorption. The small intestine, as we said before, consists of duodenum and ileum. All the absorption of the digested food mostly occur in the ileum. Glucose and amino acid pass into the bloodstream, fatty acid and the glycerol pass into the lacteal and then to the lacteals and then to the lymphatic system. So if we want to summarize the route of digestive food, as we told before, the uptake and use of food is called assimilation. So the small molecules that have been already digested enter the epithelial cells. Soon from the epithelial cells it will move to capillaries in the villus, soon to the vein, soon to the hepatic portal vein which carry blood to the liver, then to the liver, then to the general blood circulation. So liver acts here as a stage for detoxification of the food coming from the intestine. You can see as well that capillaries, some of the fatty acid absorbed and glycerol enters the capillaries in the villus and have the same routes as the other small molecules. While most of the proportion of fatty acid and the glycerol go through the lacteal and to the lymphatic system and soon will emerge into the general blood circulation. If you look closer again to the structure of villi, as I told you before, you have blood coming from the aorta, and then you have blood to hepatic portal vein. And you have the lymphatic system, which collect most of the fats, most of the glycerol and the fatty acid formed. And these are the route of the absorption of the smaller molecules. So we have three major smaller molecules that can be easily absorbed. Glucose, we have amino acids, and we have fatty acid and glycerol. So glucose and amino acid will pass into the capillaries and soon collected by the vein to hepatic portal vein to the liver to the general circulation. While the fatty acid and the glycerol, most of it, around 90 or to 95%, will pass through lacteal and then to the lymphatic system, while very small proportion will pass through the blood vessels. You can see that arrows below shows what is the kind of diffusion. So the big one, the dark big arrow shows that is a mass flow. A lot of substances are moving. While the white big arrow, it talks about active transport. That means that molecules are transported using energy. While the small arrow, thin arrow, it is the diffusion down a concentration gradient. That's meaning it's moving from a high concentration into low concentration. So how the small intestine is adapted for absorbing digestive nutrients. So we'll talk about the feature that make the intestine, a small intestine, a very good structure for absorbing digestive material, and how this help absorption take place. Actually, the stomach, small intestine is very long, about 5 meters in adults, and this gives plenty of time for digestion to be completed and for this digested food to be absorbed as it slowly passes through the intestine. It has villi. Each villi is covered by a lot of smaller projection called microvilli, and this of course will increase the surface area for absorption. The larger the surface area, the faster the nutrient can be absorbed. Villi as well contain blood capillaries, and these blood capillaries will aid the movement of these monosaccharide, amino acids, water and mineral vitamins and some fats to pass into blood and to be taken to the liver and then around the body. The villi as well contain lacteal, which is a part of the lymphatic system, which is the fats to be absorbed into lacteal. The villi has walls only 1 cell thick. It's only 1 cell thick, and this enables the digested nutrient to be easily cross the wall of the intestine to reach blood capillaries and lacteal. In this slide, we'll see how the body controls the blood glucose level, the normal blood glucose level. The normal blood glucose level is around 70 to 110 milligram glucose per 100 ml. So if blood glucose rise, such as after eating, the beta cells of pancreas start to secrete insulin. This insulin promotes two things. Insulin enhances the transport of glucose into body cells and stimulates as well the liver to store glucose as glycogen. So that leads to blood glucose levels fall. What happens if the other way around occurs? So if the blood glucose level falls, such as after fasting, the alpha cells of pancreas secrete another hormone which is called the glucagon, and the glucagon have an antagonistic effect to insulin. This glucagon will promote the breakdown of glycogen in the liver to release glucose into the blood, and this will raise the glucose level again to reach the normal level of glucose.